All right, so today, I actually designed our workout today. It's an endurance workout, so one of our running days. The idea was we had some cones set up, and it was all about clocks. So basically, if you think like, your clocks here, um, is you would start with quarters. So, boom, boom, bottom. So 12, 3, 6, 9, 12, back to 12. Idea was 12 of those. Um, then you would do, or sorry, that was the next one. It was 12 of just front back. So sprinting, backpedaling, sprinting, backpedaling. You did 12 of those. Then you would do 6 of the quarters. And then finally 12 of, or not 12, Jesus Christ, 3 of each hour, basically, and all the way around. Roughly a minute and a half break between each set. But it went pretty well. But today we talked a little bit about DNA, kind of how that works. So DNA obviously is our genes. It's 6 nucleotides, 3 base pairs, codon as a whole. So we have 2 main ways that we talk about when we're talking about DNA, or how we make more DNA, more proteins. You're going to see transcription, which is all about writing the proteins. So you're not quite making them yet, you're just laying down the framework for them to be made. So how this is going to be done is you have polymerase, Pol2, which is going to come, kind of sits on DNA and kind of splits it. Because as we know, DNA is that double helix, so polymerase is going to take it, make it into those 2 individual strands, kind of unzip it, so that you can, matching codons can come and read that DNA and make half of a DNA strand, which is basically called RNA. That potentially, we'll get to a little bit later, but that potentially will then go into translation, where the little RNA will go to a ribosome, and then a set of steps happen, and then boom, new protein is made. But there's actually this really cool kind of thing that happens where we can have transcription and not have translation occur. And how this kind of happens is we can have our body telling itself like, hey, like we need to make all of this protein, like we're low, for whatever reason. But if we do not have the amino acids and the protein present to actually make them, then it's just not going to happen. So you can have tons of transcription happen without any translation happening. But we can actually see as there are some things that boost transcription levels, like transcription signalers are going to increase it. There's some, I believe, some other chemicals do as well. But yeah, so the big takeaway is that you can have one without the other, specifically transcription without translation.